Hello, everyone. My name is Kellen Cavagnero, and the title of my talk today is Cyclic Di-GMP Induces Sting-Dependent ILC2 to ILC1 Shift During Innate Type 2 Lung Inflammation. This work was completed in the lab of Dr. Taylor Doherty at the University of California, San Diego. Asthma has been linked to infection, both bacterial and viral, as well as cellular stress. To improve asthma treatment, it is critical to understand how these triggers impact asthma pathogenesis at a mechanistic level. To that end, our group began thinking, what do these processes have in common? Well, infection and cell stress lead to the generation of danger signals, including the cyclic dinucleotides. Our lab studies recently discovered group 2 innate lymphoid cells, or ILC2s. Lung ILC2s drive innate type 2 inflammation in mice and appear to contribute to asthma and other allergic airway diseases in humans. Importantly, how these cyclic dinucleotide danger signals regulate lung ILC2 responses is unknown. Here we investigated the role of cyclic di-GMP, or CDG, in a fungal allergen-induced asthma model in order to better understand the mechanisms linking infection and stress with asthma. This knowledge will ultimately help us better understand and treat asthma in humans. To our surprise, we found that in an alternaria-induced ILC2-driven model of innate type 2 inflammation, treating mice with CDG completely abrogated airway eosinophilia. We also noted increased levels of type 1 interferon as well as IF and gamma in the bronchoalveolar lavage of CDG-challenged mice. Because both have been reported to strongly suppress ILC2s, we next looked at how CDG affects ILC2s in vivo. We found that CDG challenge decreased the levels of type 2 cytokine producing ILC2s as well as ILC2 expression of GATA3, the master type 2 cytokine transcriptional regulator. Interestingly, we noticed a concomitant expansion of IF and gamma positive ILC1s and an increased expression of TBET, the master type 1 cytokine transcription factor. These cells are critical for combating intracellular pathogens like viruses. We did additional experiments that suggest innate lymphoid cells are the major IF and gamma producers in this model. These findings may help explain the mixed inflammatory phenotypes seen in a subset of human asthmatics. So, how exactly does CDG suppress ILC2s and type 2 inflammation? Well, one of CDG's receptors is STING, the stimulator of interferon genes, encoded by the TMEM173 gene. Because we saw increased levels of interferons in the BAL, we tested the dependence of STING in our model. Suppression of type 2 inflammation and ILC2 responses was completely STING dependent. Given that STING has been shown to drive the production of type 1 interferons, which again were increased in the BAL of CDG challenged mice, we tested whether type 1 interferon signaling was required in our model using IFNAR1 knockout mice. Removal of IFNAR1 completely abrogated the effect of CDG. Taken together, our work suggests the following model. Alternaria, which induces epithelial release of IL-33, leads to the activation of ILC2s and eosinophilia, and CDD danger signals bind to STING and drive the production of type 1 interferons that inhibit ILC2s and downstream eosinophilia. Finally, the increased levels of type 1 interferons and the novel ILC2 to ILC1 shift we observed suggest that CDG may be a potential local respiratory therapeutic for treatment of infectious diseases wherein protective interferon responses are inhibited, such as in SARS-CoV-2 infection and COVID-19. Thank you all for your attention, and thank you to Quad AI for the opportunity to speak today.